Hi everyone. I'm here to discuss my work which deals with the prediction of binding affinities and understanding the mechanism of inhibition of FDA-approved compounds on hepatic transporters BSEP and MRP2. Now, for a metabolically dynamic organ like liver, membrane-embedded transporter proteins play a crucial role in facilitating the influx and efflux of a range of exogenous and endogenous compounds from the hepatocytes. These act as critical determinants in drug distribution, metabolism, and biliary secretion. Owing to these central functions, any kind of interference with the functional transport processes can lead to the intracellular accumulations of toxins, which can further lead to cell damage and cholestasis, often together termed as drug-induced liver injury or, in short, DILI. Of note, the bile salt exporter pump BSEP is considered the primary possible transporter-mediated DILI-inducing culprit, and MRP3, MRP4, and MRP2 proteins follow closely on the list of transporters that are most likely to be affected by drug-induced cholestasis. In this work, we have mainly focused our attention on the canalicular efflux transporters BSEP and MRP2 because clinical reports have presently mentioned that around 30% of the clinical drug-induced liver injury cases are being primarily caused by cholestasis as a result of BSEP inhibition, with concomitant observations of conjugated hyperbilirubinemia as a result of MRP2 inhibition. Now, while several experimental studies have confirmed the potential drug inhibitors of BSEP, the lack of an experimentally validated MRP2 structure in the protein data bank limits the scope of extensive ligand binding studies with MRP2. Consequently, in silico approaches in combination with homology modeling studies have revealed the identity of several inhibitors which are possible inhibitors of both BSEP and MRP2, which favor a comparative analysis. Furthermore, computer-aided drug designing or CAD approaches are important for reducing the use of animals in vivo testing, for designing of safer drugs, and for repositioning of the already known drugs in the market. To briefly discuss the experimental strategy that we have obtained or that we have adopted in this work is downloading of the VSEP structure from protein data bank and modeling the MRP2 structure using homology modeling approach via Swiss model. And the possible inhibitors of both the proteins have been obtained from the online repository PubChem, and these were around 100 in number. This was followed by target and ligand preparation, and the selected compounds were later on docked using Autodock Vena, and which was subsequently followed by post-docking analysis using Discovery Studio Visualizer. And we have additionally also performed toxicity predictions of the lead compounds using Vega. Now, moving on to the results section. Around top 14 compounds with most negative binding energies were listed and ranked accordingly. And from our results, we can see that around 6 compounds are common for both BSEP and MRP2, and these were staurosporine, zafilicus, clofazimine, itraconazole, tolvaptan, and glypedol. Strikingly, staurosporine appears to have the most negative binding energy for the BSEP as well as MRP2 protein. And post-docking analysis has revealed that the docked complexes had mostly pi-pi interactions and edge-bonding interactions in between staurosporine and the BSEP and MRP2 target proteins, respectively. Similar kinds of interactions were obtained with the other lead compounds and the target proteins as well. And toxicity prediction with staurosporine showed that staurosporine is mutagenic and hepatotoxic in nature. However, it was not carcinogenic in nature. Similar interactions and similar results of toxicity predictions were obtained for the other compounds. Now, moving on to the conclusion of our study, it was a general observation among the 100 screen compounds that compounds having in general lower IC50 values for the two proteins had most negative binding energy for both the proteins. Also, the dog complexes were dominated by both ionic and non-ionic interactions, which further dictate the best fit of the lead compound at the protein's active site. Such in silico studies are therefore helpful in not only reducing the overall drugs to be screened in the entire process, however, they also bring down the overall cost, the overall manual human efforts that are behind the development of a successful drug for a particular disease. Studying these hepatic transport is extremely crucial because liver, as we all know, is crucial and central to all kind of drug metabolism, and any kind of perturbation with liver enzymes or liver transporters can lead to serious consequence inside our system. It is therefore important that these preliminary findings of the in silico studies are validated down the line with in vivo studies, all in vitro and in vivo studies, although limited in number, so that our findings are standing true in an actual biological system. With this, as I'm about to conclude my talk, I would like to thank my mentor, Dr. Ashwani Sharma, for providing me with this beautiful opportunity to present my work in the third Women in Bioinformatics and LA conference. I'm extremely grateful, and thank you all for your patience hearing.